Hi, everybody. I'm Margie Weiman. It looks like on the slide I'm from City of Hope, but I'm not. I'm actually from UCLA. I'm here to talk about non-cellular and in vivo gene therapy. I'm going to do a quick show of hands. How many of you here in this room have taken care of a patient that's gotten non-cellular or in vivo gene therapy? And if you don't know what I'm talking about, you probably haven't done it. So show of hands for people who've done it. In my group, you better raise your hands. Okay, so not so many, right? So this is going to be a new topic. Dr. Cohn luckily talked about a little bit about it. And he went over some of the CRISPR stuff, so that's great because I don't really know that kind of stuff. So I'm glad he went through that. So most of what you've been talking about in this conference has been in this middle, in this column here in the blue and the purple. The CAR T's, the purple, the blue is the stem cell, and when the stem cell and gene therapy come together, that's your section over there on the left. But I'm talking about this little yellow box over there on the far left that is just gene therapy and does not involve any cells. So in vivo versus ex vivo. Now, ex vivo is, again, what we've been talking about. Cells come out of the body, cells get manipulated, cells go back in the body. In vivo, you're actually having the gene wrapped in a viral vector, and that's being administered directly into the targeted tissue or the bloodstream. The history of the tracks, oopsie, I broke it. The history tracks along with the cellular therapy, but I will tell you, fun fact, in 2012, the first FDA approval for in vivo gene therapy happened. It was for a congenital hyperlipidemia disease, and they brought it all the way to market, and they treated one patient. It's called Glybera, and then it was so rare, there were no other patients to treat, so they took it off market. I mean, there were probably other patients to treat, but it wasn't financially feasible to keep it available. So the current FDA-approved in vivo gene therapy products are up here on the screen. There's 6 of them. The first one was of the current batch was approved in 2017, Luxturna for hereditary retinal dystrophy. And then 2 more approved last year in June, Elevidys and Roctavian for Duchenne muscular dystrophy and severe hemophilia. I'm mostly going to focus on these FDA-approved products. I'm not going to go into the clinical trials. I will tell you that the clinical trials have expanded exponentially, over 3,000 the last count in 2023. So types of in vivo gene therapy, and again, Dr. Cohn touched on this also, but we have to talk about the target cell. So is it somatic or is it germline? Somatic cells are the ones that do not produce sperm or eggs. Effects will not be passed on to offspring. Germline are target cells that are producing sperm and eggs, and the effects could be passed on theoretically to subsequent generations. As of today, we are only discussing somatic cells. There's a big ethical issue, obviously, with doing manipulations of germline cells, so only somatic cells at this point. And also, when you're talking about gene therapy, you need to talk about what's being manipulated, what gene, and how is it being manipulated. So gene augmentation or addition, you can add a functional version of a lost or malfunctioning gene. You could also silence a gene where it decreases the amount of a specific protein that's being made by the target cell. And then there's gene editing, which Dr. Cohn went into, again, thank God, so I don't have to. Now, where is the gene targeted? So is it going to be in the DNA or is it going to be in the RNA? And for different diseases, it'll work better for different locations. So in DNA, the transduction actually occurs in the cell nucleus. It's a greater chance of it being permanent, which is when you see just a single infusion only being done. So one and done. RNA is when the transduction is going to occur in the cytoplasm with the RNA molecule. And different types of RNA can be targeted. It can be microRNA, small interfering RNA, or antisense oligonucleotides. There's actually a fair number of FDA drugs already approved that are antisense oligonucleotides, ASOs. My talk today is really going to be only focusing on the DNA targeted genes. Now, when we talk about in vivo gene therapy, we need to talk about vectors. So vectors are the substance that presents the new gene to the cell into which it will be incorporated. You can think of it as the suitcase for the gene. There's viral vectors and there's non-viral vectors. The types of viral vectors include adenovirus, adeno-associated virus, and lentivirus or retroviruses. And those lentiviruses are used often in cellular gene therapy. So, for example, Dr. Cohn talked about those. Viruses evolved to enter cells efficiently. That's what they do. That's how they propagate. And so they are really good at taking the gene and presenting it to the cell that you want it to get into. But they do cause an immune response. So non-viral vectors are a little bit better sometimes because they will lack the ability to provoke an immune response and include things like lipid nanoparticles, LNPs, or plasmids. The other issue with viral vectors is non-viral vectors tend to be a smaller suitcase. You could think of them as the carry-on size, whereas the viral vectors are the ones you take on the 2 or 3 week vacations. Most FDA-approved in vivo gene therapy products use adeno-associated viral vectors. These typically require other helper viruses to infect the cells, like herpes virus or adenovirus. They do not cause disease in humans. So they infect cells, but they're not pathogenic. So you're not going to get cold symptoms or other symptoms from an AAV vector, adeno-associated viral vector, AAV. They tend to be less immunogenic than the adenoviral, which is why they've come into favor. As I said, they're a smaller size as a suitcase. And the awesome thing about AAV vectors is that they have different serotypes that target different types of cells. So AAV8 goes for skeletal muscles, cardiac myocytes, retinal cells. It only goes for those particular cell types, whereas AAV3 goes for inner ear cells and hepatocytes. The vectors in the current FDA-approved products you can see are in this table. So Luxturna is the vector is the AAV2 vector, and it's targeting retinal cells. Elevidys is actually using an AAV that's modified from a rhesus monkey, and that targets muscle cells. The 2 hemophilia drugs or gene therapies are using AAV5 viral vectors, and they target hepatocytes. Now, when we talk about in vivo gene therapy, we need to talk about challenges. There's a young man named Jesse Gelsinger. He has ornithine transcarbamylase deficiency. And in 1999, he volunteered to be in a clinical trial, and he was dosed with an adenoviral vector where he received, he wasn't the first person on his dose cohort. He wasn't even getting the highest dose, but for some reason, he got an adenoviral vector and he had an overwhelming immune response. He ended up dying about 98 hours after he received his gene therapy product. This really caused a pause in the in how everybody thought about gene therapy. Everything was sort of a full stop while everybody thought about, well, what are we doing here? What's safe? What's not safe? We really need to look at these viral vectors a little bit more. So much is unknown about the mechanism of gene therapy. There are several adverse reactions to gene therapy that you need to know about if you're giving these. Immune responses to AAVs. So humans injected with large doses can mount a B or T cell response against the capsid or even the gene therapy product. Complement responses, the complement system is the body's natural defense against viruses. But when you're getting an overwhelming load of AAV, you can get a thrombotic microangiopathy. So sometimes patients that are getting this will get plasmapheresis or eculizumab, which is a monoclonal antibody which can tamp down the complement system. And then probably the biggest concern is when you give this product, but there's failed transduction, so it just doesn't take. And this can happen due to neutralizing antibodies or T cell response. In this graph here, the red line, so this is a patient who got a factor 9 gene therapy, and you can see the red line is factor 9 production. It spikes up to week 2, but then gradually starts to come down, and you can see the blue and the green line are ALT and AST, and it indicates the liver damage that's starting to occur, and the transduction is being sort of fails. It's failing, and the factor 9 production just decreases back down to 0, and so that's an example of a failed transduction. Other adverse events that can happen with AAV vectors include infusion reactions, which we've talked about ad nauseum, and which you can also get nauseum. Hepatotoxicity, which can occur due to a vector, especially if it's targeting hepatocytes. The dorsal root ganglia toxicity, if the in vivo gene therapy is being given intrathecally, they can get sort of symptoms of peripheral pain or numbness and deficits in sensory perception. Myocarditis can occur, especially if you're using a vector that's targeting skeletal muscle cells. So oftentimes patients will get troponin levels measured serially post-infusion. And then the FDA has recently said that patients that are getting AAV vector products need to have a minimum of 5-year follow-up for insertional mutagenesis. The other thing we need to talk about with viral vectors and in vivo gene therapy is viral shedding. So post-therapy, what will happen is the vector from the patient gets shed for a few days to a few weeks post-infusion. And, you know, when clinical trials are going on with these new viral vectors, they will sometimes look for this and sometimes they don't. It's not required to be reported. But what can happen is that if you are exposed to the viral vector, not having even gotten it yourself, but you're exposed to it, you can seroconvert. The non-affected person can seroconvert, which means you can develop antibodies to the viral vector. Why is this a problem? Well, it's mainly a problem for people who have genetic diseases in their family. So a patient's family, a family with 2 boys who have Duchenne muscular dystrophy, for example. Sibling A gets the gene therapy product, and if those 2 boys aren't separated, then the viral shedding that happens with sibling A can cause sibling B to seroconvert, develop antibodies to the viral vector, and then when it comes time for sibling B's turn to have the gene therapy, they can't receive it because they already have antibodies and transduction will fail. Does that make sense? Yeah. Also, an issue is the cost of these gene therapies. You can see that some of them are some of the most costly in the world. Duchenne muscular dystrophy, that particular infusion, just a one-time infusion, $3.2 million. Can you imagine being the nurse holding that bag and that tubing and God forbid something leaks? So they estimate that the annual spending on gene therapies are going to average approximately $20.4 billion. And they're saying that the cost and funding of Medicare and Medicaid is going to need to be increased to help meet this demand. But the pharmaceutical companies will say to you, well, hey, gene therapy should be considered more cost effective than cellular gene therapies because there's no lymphodepletion, there's no apheresis, there's no manipulation of cells. So I'm not going to shill for them, but who knows? If you're a nurse and you're going to be administering in vivo gene therapy, you need to know the characteristics of what it is you're giving. What's the disease? What's the administration route? What's the vector? What side effects? How do you get this information? Well, sometimes pharmaceutical reps will come in and give you guys an in-service and they'll get you all tidy up and so you know exactly what you're doing. Sometimes you don't have those resources. I've actually found the FDA package insert to be a really good place to look. It's all available online too. Another thing is that you need to know when you're giving viral gene therapy, in vivo gene therapy, is that it's dosed by vector genomes or viral genomes, VG per kilogram. It can be a set dose or it could be weight-based. And the number of viral capsids or the VG that they prescribe is that what they think is going to be required to deliver a dose that will result in transduction. Targeted tissues, so when it's given directly into an eye or directly into the muscle, those are going to be lower. doses that are given systemically are going to be higher. The PPE for in vivo gene therapy, I will tell you that there are lots and lots of laboratory guidelines when they're doing bench research on this product. I will tell you there are no clinical guidelines. So NIOSH, they're the authority that tells us what drugs are hazardous. They do not include gene therapy in any of their lists. So the last list came out in 2016. New list was due in 2023. It's not out yet, but it's not going to be included. So you can't look for this information there. And then biosafety information is not always included in the package inserts. What I have up here is what I found on what different pharmaceuticals will say is required. In some cases, it's just bagging up waste. In some cases, if you're pregnant, you shouldn't care for these patients. It's going to be different depending on what the viral vector is. So I will tell you at UCLA on our infusion days, when we're doing in vivo gene therapy, we prepare the room. We put a sign on the door. We sharps containers in the room, the biohazard waste containers in the room. Only the nurse and the subject is in the room, plus or minus a parent, and no eating or drinking during administration. Then the nurse has a full PPE, just as they would for chemo, and then post-administration, the tubing, the bag, everything is put into the red biohazard waste bag, taken to medical waste area, and the room gets terminally cleaned. What happens after that is sort of a point of contention or discussion, perhaps is a better word for it. Nationwide Children's in Ohio has published a number of interesting articles about whether or not patients who receive in vivo gene therapy need to be on contact precautions. They have a system where they will intake any new gene therapies that they give in their system. They go through pharmacy and therapeutics, and they have infectious disease people who assess whether or not contact precautions are required for the particular viral vector that's being delivered. Not all places have that. UCLA doesn't. There's only so many battles I can fight, I suppose. But what Nationwide does is they put contact precautions for everybody for 30 days post-administration, and then also they identify in the EMR when these patients need contact precautions. So everybody in the health system knows. It's a really interesting, nice way of doing things. Anyway, that is my speech, my talk. I'm available for questions later.